Good morning, everyone. Thank you for joining both in person and online here in the Zoom meeting. My name is Tomas. I'm the technical sales representative for IMARIS here in the Tennessee state and the rest of the south east of the United States. Maybe some of you know me or have met me at some of the previous workshops here at Vanderbilt. But for those of you who don't know me, I'm half German, half Peruvian. So my accent is German. So just want to get it out of the way right away. And obviously my last name, if you see on the screen here, it's not very German. So that's my Latino part. So that's the Peruvian part. I did my PhD in Germany in confocal microscopy and lots of image processing and analysis. So I have experience from commercial software over to all of the open source software repositories and everything in between, like Python, Java, MATLAB, and all the fun stuff. So if you have any questions, now is the good time definitely to have me here and just ask away all your questions, and especially in the one-on-one sessions this afternoon, we'll dig deeper on your own data and try to find good ways to solve your image analysis problems. So I do like to start with this brief PowerPoint presentation. I do want to show a complete workflow as well here, but I do like to start with this slide that you see here. You know, what are your 3D microscopy goals? This also applies to 2D. Okay, most people know IMARIS as a 3D processing and analysis software, but it's also working with 2D. And my question is, okay, so you have this nice microscopes here at the facility or the lab or where else, you know, you get those nice images. What do you want to do with those images? Okay. On one hand, of course, you want to have stunning pictures for presentation or for publication, you know. In terms of visualization, you know, you have the probabilities of doing 2D, 3D, maximum intensity projection, all those things, or even make animations and movies where you can, in a presentation, you know, zoom in through your data set, fly through different objects and those kind of things. And this is all great and nice, but on the other hand, you know, what people want to do on top of visualization is quantification, you know, extract meaningful data, meaningful numbers out of your data set. So this is, so to say, the heart of the IMARIS software. This is what I'm going to talk about mostly. This presentation is just an introduction to show you all the capabilities of IMARIS. I mean, it's especially geared towards beginners, new users, or people who have maybe used it before but are not, you know, experts in IMARIS. And I do want to just build up on this and then show a little bit of more advanced features in the second half of the presentation, but everything builds up on it, even if you haven't used IMARIS or just a beginner. I mean, it should be not a problem to follow along here. Okay. And if you have used IMARIS, then the beginning will be a bit of repetition, but I'm going to throw in some new things here and there, especially with new features and new things that happened in new releases that you may not be aware of. So in terms of quantification, this is like a little workflow that I would like to show hands-on later after the 20 minutes or so PowerPoint presentation, where we can extract, you know, cell count. We can see how much are the distances between all different objects in space and get some graphs and things like this, okay? On top of this, you can also do tracking, so meaning live cell imaging for the analysis. You know, there's different ways to label this, but essentially it's like a time series, the movie, where you can track objects over time and get dividing events of cells, maybe track speed, acceleration of objects, those kind of things. Okay. So typical workflow looks like this here. So IMARIS is a post-acquisition analysis software. Post-acquisition means that you acquire first your images with the microscopes you have available. And this is the first step. And then you import the files into the IMARIS software. You do some pre-processing is an optional step. It's a cleanup step. Okay, just removal of artifacts, you know, noise reduction. Your image may be already perfect and nice and awesome. You don't need to do it, but we all know life is not perfect, and we always have some stuff going on in our images, and maybe even some in the time series we have some you know, somebody bumped our microscope and we have some drift or something going on. So these are all things that we can address here in the pre-processing step. Then in terms of visualization, it's like the rotation, the interaction with 3D environment to have a look at what you have available. And then in the analysis portion, it's like the heart of the IMARIS software. This is where we do extract the features of interest. This is what we call segmentation. we segment the objects, and then we extract statistics, volume distributions, localization analysis, overlap percentages, those kind of things. And then we can also use the last step is classification interpretation to see what do those numbers actually mean. Okay. And then we can also automate everything. We don't have to reinvent the wheel again. So I have a slide about each of those to dig a little bit deeper, because this is like a general flow. You may skip 1 or 2 steps here, or maybe you'll have an additional step like an animation or something, but this is just a general overview of what you typically encounter if you use IMARIS. So starting with the acquisition, IMARIS is an agnostic software, so it means a wide variety of imaging modalities can be used with IMARIS software. Most people know it as a fluorescence microscopy and image analysis software, which is the case. Like 80% of our users use it for fluorescent microscopy or any type of optical microscopy. But we also work with electron microscopy, with MRI, CT, micro CT, slide scanner, immunohistochemistry slides. So any 2D, 3D, and 4D image data can be input into the IMARIS software and worked with. Especially now with the advancement of machine learning techniques, it's much easier and more streamlined to work with non-fluorescent images in IMARIS. So this is the acquisition, okay, in terms of modalities. That now in terms of, vendors or brands of microscope and things like this. So you can import a wide variety of data into IMARIS, because we do require a conversion into the IMS file format to have a more seamless experience, to make it smoother, to make the algorithms work faster, and you have a seamless, zoom in, rotation, like interaction in a 3D environment. So we natively support all the big 4 vendors like Zeiss, Nikon, Leica, Olympus. You can get your CZI size file, for example, and simply convert it with a file converter, or double-clicking on it in the IMARIS arena, and it converts to the IMS file format. Also very popular is a TIFF stack, or any OME TIFF for larger data. But we have a wide variety of vendor file formats that we support as well. And we also have bioformats in case, there's something that is not directly supported. So now, once we convert our file to the IMS file format, we can start with the pre-processing. And I mentioned it's an optional step, but to complete the picture, we do have IMARIS stitching capabilities. So we can stitch if you have want to capture a larger field of view, you have different tiles and you want to stitch it, make a montage or make just one large image out of it. I know you may already have access to like the size software, sense software, where they have stitching capabilities, but this is mainly an additional use to not block the acquisition workstation for your stitching needs. So you can do it offline on a separate IMARIS workstation. Same goes for deconvolution. IMARIS has deconvolution capabilities, but you may already have this from your microscopy software. But nonetheless, you can also use it for offline deconvolution. Deconvolution is also batchable, meaning you can just point IMARIS towards a folder where you have 100 images and batch deconvolve all 100 images while you take a coffee break or lunch break, for example. So this is deconvolution, but then we have, as I mentioned earlier, we have a wide variety of noise reduction filters, artifact removal, like a regular median filter, for example, for salt and pepper noise, or background subtraction is one of our most popular filters. And I'm going to talk about it a little bit more in detail when I go into the hands-on session after the PowerPoint presentation, where I can show what effects do we have with background subtraction and how can it help us, get a better pre-processed image to help us with the segmentation results. Any questions so far about pre-processing anything? Everything clear? Okay. So coming to visualization, as I mentioned, this is like the interaction in a 3D environment, but most people don't know that we offer 4 different rendering modes. Okay. So the default is the MIP, the maximum intensity projection that you see on the left. This is where we typically, where I interact in a 3D environment, where I do my analysis, I do all my process pipeline, I work in the MIP, and I can take snapshots and images in the maximum intensity projection as well. It's not a problem. But as you know, or if you may be familiar, the maximum intensity projection, even though it's a 3D mode where we interact in the environment, you cannot create as much of a depth and you cannot always tell if a structure is behind another structure or one structure is in front of another structure. So therefore, we offer also blend, normal shading, and shadow projection modes, which can enhance, the depth, the opacity, and create more of a 3D effect for a publication, for example, or a presentation image. Okay, so we offer those. The blend is definitely the most popular, I would say, together with the maximum intensity projection. But we have other tools as well. So coming now to the analysis part, as I mentioned, this is, the core functionality of IMARIS. So IMARIS is an object-based image analysis software. What it means is that we have objects, and depending on what your object or your data looks like, and depending on what statistics you would like to extract, you will be using one of 4 objects. So we have 4 main objects in IMARIS, and everything boils down to those 4. I'm going to talk about these 4 objects in the next slides. As it says here in the 3rd bullet point, it's a simple step-by-step creation process. What it means is, for example, when you remember when you last installed a new software, you get this, for example, this box that tells you, hey, do you want to create a desktop icon? You check the box, click next, and it guides you through step-by-step wizard. There's the same principle here. We have some checkboxes, some numbers you type in, and then typically you have 4 to 6, 7 steps to get to this segmentation result. The 4 objects that we have in IMARIS are spots, surfaces, cells, and filaments. So everything boils down to those 4. It really depends, A, what does your image or your structure of interest in your image look like? And B, what are the statistics that you would like to extract from your data set? Okay, because sometimes you may have to use 2 of those. Sometimes it doesn't look very spherical, but spots will still help you. So it really matters, and I'm going to discuss this more in detail, but it really matters, what statistics you would like to extract from the data set. So starting with spots, okay, I'm going to start with the first object, which is spots, and then build my way up. As it says here, to detect objects based on local maxima in 3D, okay? So it is used if you have, for example, in this case, like little nuclei or some cells, that are fluorescent, you want to detect those. And it's oftentimes used for cell counting or object counting, and also for tracking, okay? As it is very fast. So it is the least, the object that uses the least amount of resources on your workstation. Okay, so you can imagine, microscopy data is not getting smaller anytime soon. It's actually the opposite. It's getting a lot bigger and bigger. Especially you guys, if you're using light sheet data, you probably know it's you're out of the megabyte range by far and going into the very high gigabytes to even terabyte range. So you will have will need to have a dedicated workstation to work with IMARIS in most cases. I mean, depending on the size of your data, but again, as time soon, so you will have you will need a more powerful workstation. If you don't have the resources, or you have your data that's very, very large, but you are only interested, in tracking speed, for example, or displacement, you can use the spots because it doesn't take as much computational resources. So it means you can get to the result faster, even with a less powerful workstation, or even if your data is really, really, really large. So people use it here for position, count, position count, intensity analysis. Nearest neighbors gives you an indication how densely packed are objects in some regions versus others. As an example here, we have this Drosophila egg chamber. The researcher here, so in this case, they wanted to count the number of the small nuclei, those guys here at the top right, not the large one, like those small guys, and measure the distance from each of those towards the tip, in this case, towards the end of here, okay? So with IMARIS, we can place the little spots here, the little spheres. This is our spot object into each of those, so we get a count. And then also we can get intensity analysis, we can get, nearest neighbor distribution, but also we can get the distance from each of those to a specified point, in this case, the tip here. So this is spots. Going to surfaces, as you can see, it rearranges here. So surfaces, I like to say, can do everything that the spots can do, but more, okay? So now we're going into the morphology. So with a spot, you place a little sphere, but most objects in biological objects are not perfect spheres. So they have an actual shape. So we use surfaces to actually capture the real shape of objects. Okay, and this way we can get morphology. Now we can go into volume analysis, surface area, sphericity, how round or spherical are our objects, tracking, overlap, like how much overlap do I have between surface A and surface B, those kind of things we can do. So now we wanna, this guy here or this girl, this researcher, they wanna detect larger nuclei boundaries and measure shape and size. So those larger nuclei, we wanna get the actual shape and size. So we place a surface. So you see now it's not a sphere anymore, it's the actual surface. And measure shape and size, meaning the volume, surface area, how round is each of those nuclei, like the sphericity. And this is what we can do with the surfaces. So next up here, surface and spots are big data ready. Okay, so what it means is, as you see here, we can detect, track, and interactively render millions of spots, okay, or surfaces for that matter. So the limitation is not the software, it is the hardware that you have, okay? And this is a little bit older slide now, because also since the last release, filament is also big data ready, okay? So we can now also use filament tracing for example, light sheet data without you having to wait 5 days for a result, okay? So this is also like one of the new features I would like to talk about today. But definitely spots and surfaces are big data ready, so you can work with it on very large data as well. Questions about spots and surfaces? These are like the two main building blocks that you will see quite a bit. Any questions? So if your questions on the Zoom, just also type your question in the chat, and then Jenny will be able to read those questions as well. So going to the next object, okay? So this is cell and then filaments. So as I said, spots and surfaces are the two more basic ones, okay? So that you will be using quite a lot of times. And then cells and filaments are more specific ones, like you will be using more for dedicated and specific workflows, okay? So the cell object is to detect cells and their subcellular components, okay? What it means, the key difference between just having spots and surfaces and cells is to get statistics on a per cell basis or on a per object basis. It doesn't need to be a cell. Just to give you an example to make this clear, so on the right-hand side, you see we have 4 cells here, those guys here. And then you see like each of them has their nuclei, the cell boundary, like from a membrane stain, for example. And then you see vesicles in there or different foci. You see those in red here. So just using spots and surfaces, you can tell, okay, we have 4 cells and we have 200 of those vesicles, but you don't know how many vesicles per cell, okay? The cell object gives you, okay, cell A has 25 of those, cell B has 30 of those, and so on and so forth, okay? You see, you may be able to get away if you just have 4 cells or maybe 2 or 3. But imagine having 200 cells and doing it on 200 images. Okay, so it's going to be very tedious. So we want to automate this. The cell object can spit out the statistics right away. Okay. And you can also use this for tracking and a variety of different other statistics, but mainly this is used to use as a compartmentalization tool. Okay. So this is the cell object. Then coming to the last one here, this is the filament. There's one question. How large are the data files for transferring spots and surface images from one computer to another? Are dedicated USBs required to do so? It depends on your own data, how large it is, because we typically save the whole data plus the spots on it. I mean, it is possible to just export spot data. Okay, you can export it in the very simplest of way as an Excel sheet. So then it's literally no space at all. So you can have the coordinates of where things located in space and all the statistics in the Excel sheet. Or if you want to have the spot data itself, you may be able to open it in a different software, you have different bridges here. And just the spots typically should be very small, so definitely in the megabyte range. But if you have like a, let's say 10 gigabyte data set and you do spots, and typically it's a little bit larger, like 11, 12 gigabyte, a little bit more depending on the data, but you don't need any dedicated USBs or anything to do it. I mean, USB-C, SSD, a solid state external drive will just make your life easier because it's faster, but you can use any type of connection to transfer things. Okay, here's another question. At the software embryo, can you use spot and surface for analyzing the first couple of mitotic nuclei that are not on the surface but are located at the center of the embryo? Yes, if you can image everything in the center of the embryo, then you can track those things and you can detect dividing events. You can detect splitting events. You can get from parent to daughter cells, and you can get all of those statistics. So if you can A, see it, I mean, there's nothing blocked and you can see it, and B, it is resolved to a point that you can actually detect it. If you cannot see it with your eye, like if it's like resolved less than 1 pixel, then the software is going to have a hard time also to extract those in a good detail. So this definitely dependent on your resolution, and if you're able to image that deep, maybe you have a cleared image or something, then that's not a problem. But I mean, depending on what you're trying to do, you know, and how deep you want to image, you can do this, yes. Okay, so coming to filament tracing. Filament tracing is the fourth of the objects. So it's to detect filamentous structures, so branch, tree-like structures on one hand side. So this is everything that's like neuroscience related, like neurons, microglia, or networks on the other hand, which are like vasculature. So here we have interconnections and loops. Okay, so both of those we can detect in IMARIS, and it's now AI powered, meaning with machine learning tools since version 10.0, since January this year, to make things easier and much faster. If you've used filament tracer before, you may have experienced that it could take some time. It's a little bit slow. Now this has been changed, and it's more than 10 times faster than it used to be. Plus we have the machine learning tools, so you don't have to do any fine tuning and tweaking at the end anymore. Everything is in the wizard, and it's more batchable that way. So what's the difference between just using a regular surface maybe for your neuron and doing filament object? So if you just do a surface, as you can see here, for example, on the left hand side, the blue or the orange guy, you get the complete surface of the neuron. So you can get the total volume, and you can get the surface area, for example, or the intensity inside. But if you want to detect individual dendrites, you know, the length of individual dendrites, the thickness of individual dendrites, the intensity on each of those dendrites, then you can use the filament object, which will create a skeletonization, and you will create all of those objects to get length, diameter, branching hierarchy, how many branch points, how many terminal points do we have? Also in terms of the vasculature, how many branch points, what is the thickness and the diameter or the length of each individual segment in my vasculature. And then you can also do Shaw analysis, how complex is my neuron, how complex is my microglia, also spine and spine morphology. So these are the 4 objects, and all 4 objects you can track. You can do time analysis, meaning, for example, if you want to detect displacement or speed or acceleration of an object, you can use a spot object. If you want to detect volume changes over time, if you have some cell or something that is changing volume over time, you can use a surface to detect volume changes over time. And you can use the cell object to detect is the number of vesicles or the number of foci changing inside a cell over time. And then you can use finally the filament object to determine is the thickness or the length of the dendrites changing over time, those kind of things. Okay. And then my last slide about the analysis part is about the colocalization and object interactions. Colocalization typically also very hot topic and big topic that people would like to know and use in IMARIS. There's, you know, 2 different ways, 2 different colocalization analysis pipelines that you can do in IMARIS. On the one hand, we can use pixel-based or intensity-based colocalization, which is more the traditional colocalization based way, where we look at how many of the green pixels or voxels in 3D are overlapping with how many of the red voxels, and we can extract Menders and Pearson's coefficients amongst other statistics. And this is more, again, the traditional way. But we also have the surface-based or object-based colocalization, where you create a surface of the green channel and the surface of the red channel, and you look at the overlap percentage. Okay, so both ways are in IMARIS, and if you ask me which way is the better one, I would always recommend do both. You know, the more data you have, The more statistics you have to back up your results, the better, okay? Because colloquialization is always a sensitive topic. There's multiple papers out there talking about bias and colloquialization and whatnot. And it's impossible to reduce the bias to zero, but we have those software tools to reduce the bias as much as possible. Any questions about the analysis portion or general analysis portion so far? So now coming to classification and interpretation. So as it says here, how can I use the statistics? So I get a volume distribution, I get a distance distribution. How can I use those statistics to answer my research question? Okay. So in IMARIS, you can classify objects. So we can detect those objects and place them into discretes, user-defined bins, meaning we can color code them. We can say, hey, all the large guys are blue, the small guys are red, and everything in between is green, something like this. So we can get a visual distribution. We can also directly do 1D to 5D statistical plots in IMARIS. As I mentioned earlier, at any time you're able to export the statistics, for example, as a CSV or XML. So you can get those numbers as an Excel sheet. You can create your plots in Excel. You can use different software, Origin or any other software that you would like to create your plots. That's very flexible, but you have also the possibility to do those in IMARIS itself. And I do want to show at least two things that I like that I haven't seen like this in other software. And 5D means spatial plots in the spatial realm, and then 4D, as I mentioned earlier, is like the time plots. One form of object classification. So in terms of the classification, we have three different types of classification: two regular ones, which are 1D and 2D filter, and one machine learning based classification. So this is machine learning based classification. This is just an example. So we have this brain slide here. I think it's a brain slide or not. Slide. We have different cells here, and each of those white dots, okay, and it's very tiny, represents a different cell type. Okay, so we have just used the spot object to just maybe get a count, but just place and label every of those cells. And now we do want to label them. We do want to classify them based on the cell type. Okay, so we want to say a few of them are like cell type A, cell type B, cell type C. But there's so many of those. I mean, you could theoretically just paint and assign them, all of them manually, which is a lot of work. Or you can use machine learning classification where you paint a few of them. Like here you can see a little bit of red here, maybe just a little bit here, a little bit green. So you do a few of them and you teach the software to learn. Okay, so train and predict algorithms, where it identifies for you like what is each of those cell types without you having to manually just assign them to each cell. Just do a few and you teach it. It's an iterative process. Okay, that's the most standard way of doing machine learning. And we also have this in segmentation. So this is classification. Once you've already extracted, you can assign those versus the classification. And then we have 1D and 2D filters, as I mentioned, for classification. This is, as I mentioned earlier, very simple. You can say the small guys are green, the large guys are red, and everything in between is like blue. In this case, we have distance-based. Those guys here in blue are 0 to 10 microns away, for example, from here. Those are 10 to 20 microns away, 20 to 30 microns away, and so on and so forth. Or we can overlay two different statistics, two different filters. In this case, we have intensity, the mean intensity of channel 1, mean intensity of channel 2, overlaid here, create a heat map to see like where do we have like high expression in both channels, high intensity in channel 1 and 2 in those red areas, versus where do we have like low expression of both intensities in one channel, in one object for both channels. Spatial statistics, we can see how far objects away from other objects, and I'm going to show this real quickly in the hands-on session. So I'm going to skip over this a little bit fast here. And then finally, we have batch processing, so you don't have to reinvent the wheel every time, okay? So if you have like 200 images, they're imaged on the same microscope with the same parameters, and you have your routine, you don't have to do it all the time over and again. So you can just batch process it on all of those 100 images, okay? I wouldn't recommend just doing it once and saying, hooray, it's going to work on 1,000 images, PhD done. Nope. Just test it first on like 5, 10 images make it as robust as possible. Same with the machine learning, test it, make it as robust as possible before you apply it on 200, 1,000 images. All of those like pre-processing steps that I mentioned, like deconvolution, median filter, background subtraction, those are batchable, as well as all of the objects that I mentioned, the surfaces, spots, filaments, and cells, you can include in your batch processing routine. Okay. And then finally, you can also extend your analysis with the custom tools. So we have the native direct bridges to MATLAB, Python, Fiji, and Java. If you want to use those tools, you can include them in IMARIS or bring things from IMARIS, maybe the end back to Fiji and those kind of things. Yes.
Question on batching. When you batch, can you make it so it exports automatically into like Excel or something?
Yes. Okay. So for those guys that are online, so the question is, if we use batch processing, can it, spit out like an Excel sheet right away or something with all of the statistics? The answer is yes. So after you do batch processing, you have two options in IMARIS. There's two buttons. One is to export an Excel sheet for all of the, let's say, 100 or 200 images in one Excel sheet. You can always go and manual into each and export one per data set, but you can export a global one where it has all the statistics. And then there's a second button where you can go into Vantage, which is our plotting tool in IMARIS itself, where you can compare all of those images in one plot in IMARIS itself. So those two options are available. Yeah. And I can show this in the software itself where it's located as well. Yeah. That's it in terms of the PowerPoint presentation. I do want to show you, as I mentioned, a complete workflow in IMARIS itself. But do you guys have any questions, any additional questions, general questions about what we've seen so far? Okay, so I will like to jump into the IMARIS software directly now. You can see it here. Jenny, can you still see my IMARIS screen on the Zoom?
Yes.
Okay, awesome. So for those of you who have never used IMARIS, have never seen it, I mean, once you open it, this is the arena view. This is where you can point IMARIS towards data. So IMARIS does not see everything. You have to go and click Observe Folder here and show it where your data is located. Those are demo data sets. So you also have access to those demo data sets in case you want to play a little bit with what you see here. Oops. And I do want to just double click here on this guy here and show you. But before I start, like real quick, if you've really never seen this before, as an example here, if you have, for example, this data here, you see it's called redcells.cci. It's a data set that just came out of your size microscope, so CCI. So it has a little arrow here, this guy. This means that it has not yet been converted, so it's still a CCI file. So I simply double click on it. You see it took less than a second, and now the arrow is gone, meaning now it's an IMARIS file. So I can double click on it again, and it will open in IMARIS as an IMARIS file. Okay. Anything I do and manipulate, if I click save, override it, and anything, it's always gonna work on the IMS file. So we're not touching the CCI file anymore. Okay, so you don't see it here. It's like, where is it? Where is it gone? It's still there, but we don't want to show it because otherwise you will see everything twice. Okay, it's still there. We're not gonna change or manipulate the CCI file. Right-clicking, revert to original image. We'll just remove the CCI file again. Okay. So I will go to demo image. It's already converted. This guy here, it's called microBFS Bildschirm. And I do want to show a few things on this data set and also point out, where we have new features and things like this that you may have not seen yet. Okay. So starting with this guy here, this is the floating windows to display adjustment. Here we see our different channels. So we can turn on and off the red channel, green channel. You can change the lookup table intensity, gamma value, those kind of things, without manipulating the data. It's just how you see the data here in the 3D environment. You can change, by clicking here, you can change the pseudo color of the channel as well. So you don't have to use it green, you can do orange or something. Changing back to green. this guy here is the main window, the viewer. Here's where we can interact in the 3D environment, use standard FIR, zooming, panning, rotation. And then here on the left-hand side is where we do our analysis. So we can see little tiny icons here at the very top. Those are like my spots, surfaces, cells, and filaments. You can see it's very tiny, but these are the shortcuts. That's where we get to create our segmentation or our analysis pipeline in here, okay? So for example, if I create a spot object, so I'm clicking here, a spot object, okay, or let's start with very basic. I mean, remember I told you there's 4 objects only. You see a bunch of more icons here, and you may be asking, is there more than 4? And the others are just, for example, here in author slice, are just tools to help you in the terms of visualization. So you have an orthogonal slice, so you can slice through the data set in 3D environment, and also slice through different orientation. Okay. You're still in a 3D environment, but you can slice with a 2D slice here, author slicer. Okay. All orthogonal directions. Or you can use an oblique slicer as well, where you can slice into any arbitrary direction. Okay. You see like this. And basically you have the statistics or like the settings of everything that you have activated here at the bottom left-hand side. So for example, you can change, again, YZ plane, XZ plane, XY plane. And if I were to click on the volume, for example, here, you would see the different rendering modes that I talked about in the presentation earlier. The MIP, the blend, the normal shading, and the shadow protection. So you can just switch back and forth between those and adjust, okay? So I'm going to start very quickly. This researcher wants to have a quick cell count of the microglia cells or microglia here that are in green. And the first thing I'm going to do is I'm going to do spots to count those. I'm going to relabel this microglia. It's always good to rename things because you may have 20 spots, 20 surfaces, and you don't remember which is which. So down here at the bottom left, you can see it says step 1 out of 4. So that's the step-by-step process that I mentioned in the presentation. So it will take 4 steps for us to get the spot objects and to get our cell count, okay? So there's a few checkboxes. You can see at the beginning it may be overwhelming, but most of those things are like optional things, and you will see which ones are the most important for your data set over time. So by default, classify spots and object-to-object statistics is turned on. I typically, the first time if I do trial and error, I turn those off. If you turn both of those off, the workflow is faster. Okay, so each of those checkboxes will just take a little bit more time. Okay, this data set is very small, and my laptop is capable of handling it fast. But if you work on light sheet data and you want to do trial and error, it's always good to check those off, and just once you're done, you can later on turn them off afterwards. So classify spots is to do the classification. It's an extra step. So if I turn this off, you can see now it's just 3 steps instead of 4 as it was before on the bottom left. And then object-to-object statistics, it's relevant if you want to extract statistics between objects. Okay, like if you want to see distances between one object and another, overlap between an object and another, those kind of things. Okay, so we would like to have both of those in this data set, but if I were to do trial and error, I would just turn those off. But as I mentioned, this is a small data set, so I will leave both of them on because it's a small data set. You can always go to edit at the very top tab here, and then to image properties. And here you get an idea of the size. You know, the image size here is 230 megabytes, so it's not very big. And you can get it's X, Y, and Z. You can get the voxel size, the actual size, min and max, the data type, those kind of things. Okay. So I will head over to the next step. So here you have the little blue arrows that will guide you step by step. For me, it's 4 steps. So I'm going to the next step. Here in this step, I need to choose the channel I'm looking at. Okay, so in this case, it's a green channel because I want to extract the microglia, I want to count those. And I do need to input this estimated XY diameter. Okay, in X and Y, the diameter. It wants to know just starting point. Okay, it's not a magic number, but you want to know what area you're in. Okay, there's 2 ways to get those quick. So in the past, and what I'm still doing is you can go from 3D view at the top to slice view. So now we are in slice view, so we're going up and down the Z stack. but not in the 3D environment like before where we had the ortho slice. It's now literally just a 2D view, up and down here. And anywhere I click, I get a distance here on the right-hand side, 148 microns. So click, 54 microns. So I can go in, zoom in, and I can say click, and it's 9.9 microns, okay? So I don't have to do all of them. I can do a few of those guys, 12 microns, okay? And then I can go in and see, okay, 9.94 is actually pretty good, okay? So basically, what you want to do is for spots is something in between. So if you have 8, 10, and 12, you want to choose 10, okay? The software tries to estimate for you. So the 9.94 was an estimate. The software thought, hey, this looks likely that you would like to have. And this number is hit or miss. So sometimes it's spot on, it's super accurate, and sometimes it's completely off. So never just blindly trust it. Okay, just do a check, or maybe it's a cell, it has to be 10 microns, something like this, Just do a sanity check and before trusting the software blindly. Another way is, you can see here I have this little cube. With control and mouse wheel, I can make it bigger and smaller, and the number changes here automatically. So I can go and adjust it, to the size approximate of one of those guys, and it's the 9.94. Okay, so this is the new way to do it. So you don't have to get out. We don't want you to go slice and 3D view back and forth. Just stay in 3D view. But I still, as I mentioned, I still go to slice where I like just getting a quick distance measurement. Okay. When you zoom in, you can see actually the individual pixels by default, and I don't know why this isn't, but if you use IMARIS on the workstation by default, it is preferences display. So there's an interpolation turned on like this. So you see it looks like this, like it's kind of smooth. Okay, you don't see the individual pixels, but you can always turn those off in preferences display interpolation off. And this is like your actual data, because everything's based on pixels that you get. Okay, it's just for visualization. This for all the analysis and everything. We're using the real data, the pixel data. But most people like the smoothing because it gives a cleaner look for the result. Okay. So 9.94 is good. Background subtraction is turned on by default. I will leave it, and I will talk a little bit more about it when we go to surfaces. So let's go to the next step. And as you can see here, in the 3D environment, let's take a look. The software places the individual spots here. Okay. So I can do a check if everything looks good. Okay. Here you can do a filter, a quality filter. Default filter is a filter based on intensity. So you can say, hey, I only want to look at the really bright guys. Okay, so we lost the dimmer guys here, and we're only looking at the bright guys. So you can adjust the threshold. Okay, so this changed from A to M, this little box, from automatic to manual. So if I click here again, it goes back to automatic. This is where we capture all of those, okay, even the dim ones or the brighter ones. So if I'm happy, and I will be happy with just the default, I'll go to the last step here. This is the classification. Okay, so again, it's an optional step. I didn't have to do it, but I did want to show you all the way also with the classification. So I can choose the statistics. For example, the intensity mean in channel 2, which is the green channel, intensity mean channel 2. And I can set a threshold, everything that's less than 150 are my dimmer guys, the blue guys. And then the pink, purple guys here, those are the brighter guys. And I can add multiple more, so I can add a third one here, and you can say anything over 200 is my really bright, like my blue guys and my green guys. So you can turn off the channel. So you can get a distribution here between, dimmer than 150, 150 between 200, and brighter than 200. Okay. Again, good thing to do, housekeeping rule is just like clicking on the name and changing it from class A to, dim maybe, and green to bright, and then this to medium or something, to know what is each of those. Again, for 3, you may remember, but imagine like a month or 2 later, you're like, what was green? So it's always good to keep those in mind. And then finishing up, there's no blue arrow. It's just a double green, meaning we're done. This is the end. And at this point, we can go directly into statistics here, and we can get a total number of sports 343. So we get a count right away. Okay, and maybe a question: will this recording be made of the weight of the water? So yes, Jenny you will be making the recording available correctly to everyone. Okay, so recording will be available. Yes.
To see the dim cells. Sometimes the for that sort of coalesce whether with 3 to 4 levels, one is cells that look separate when the image is dim, look separate when you brighten it. In fact, it's more.
So you mean like if you do like this, like those are like connected then again, you mean, and this will not capture those. Yeah, so yes, you can keep some of them and treat them separately. That's a possibility. So for example, we have the filtering tool here. So if I go to filter, which is here, this tab next to the statistics. So I could add a filter and I could go into the intensity mean, for example, of channel 2, or I can go intensity max as well, the different choices. And I can go to the very bright ones, like for example, those guys, like I have to define the threshold. So let's say 170 is my threshold, okay? And then I can duplicate the section to new spots. So now I have, these are all my spots here, those, and these are my spots that are like the brighter guys. You can already see this because you see the blue guys are going away. And I did classify them. The blue guys were the dimmest ones. So now I have those already, and I can treat those separately. Like I already have those, and now I can bump this up, and then I can do another microglia, just focusing on the dim one. And then at the end, I can also merge those two into one new one to maybe try to capture the very dim ones in a separate fashion, yes. So there's a few possibilities to do this, yeah, absolutely. Let me just delete this, guys. Here's the microphone. Okay, so again, this is the result that we got. I'm deleting the filter, that 343 of the total, but we can also look at each of those individually. So if I go to the detailed statistics, that's where I spend most of my time in detail and specific values. I can look at the intensity mean of channel 2 this here. And for example, I can click on this guy and it tells me the intensity 165. And I can click on this guy and turns yellow, and then the intensity is 198. I can also go the other way around. I can say, hey, which one is 140? So I can click in the table and one of them turns yellow, this guy here, okay? Which one is 190? I click and then one turns yellow. If you don't find it, there's a zoom button here. So you can just directly zoom into this guy. So this is a way how you can do this. You can also sort from highest to lowest by clicking on the name. And you can say, hey, which one is the brightest guy? Just 2 of 240. And you can take a look at this, and yeah, this is pretty bright. So that's just a few ways to find those. In the table, you always have set 1. You see the set 1 here. It tells you the classification that we've done and that I labeled, the bright, medium, and dim. So any statistic you export, in this case, it's not it doesn't give us more information because we have the intensity, and then the classification is also based on intensity. But if I were to do like a distance-based statistic export, I will also have this medium, dim, and bright always in my table that I export the second set of statistics. So here you have the floppy disk. Okay, welcome to 1998. So you can export the single floppy disk, and you can export a single statistic. So in this case, I will export only the intensity mean of channel 2. If I were to go to the multiple, I think it's 3 or so floppy disks here, it says extract all statistics. So I will extract all the statistics to CSV, XLS, or XML. And by all the statistics, I mean literally like all of those, area, average distance to 3, 5, and 9 nearest neighbors, diameter, all of those that you can see here. You can customize this. So if you're only interested in like 10 statistics, you can turn off all the other statistics and just export those 10 that you're really interested in. Okay. One more question. Is there or some overlapping glia in the stack, will it differentiate them from each other or count one spot if there were some overlapping glia? So the spots itself, is determined on the size that we told it, like the 9.95 or like the 10. Okay, so if there's overlapping microglia, then they would have more of this, more of a length than just 10. It would be like 20 or maybe 16 or something. If two spots fit in there, it will separate them. But if you are looking to more accurately, classify or segment touching objects, then I would recommend doing a surface, because there you can split touching objects. And the surface is what we're going to do next. And I can show exactly how this works if we had overlapping glia. So any questions? Yeah.
Here, when you measure the intensity, cannot spheres, they have a little figure like this. It counts the intensity of the whole thing, or just like what it counts as a sphere?
Yep. Good question. So the question here from the audience is, we have the microglia, they have the processes and extensions in here. They're not obviously perfect spheres, but if we have the sphere here and we look at the intensity, what intensity is measured actually? So it's only the intensity into consideration. It's really just the center of the sphere. Okay, so this is you have to be careful in this case. Oh, your connection is unstable. You have to be careful again with what you do. So this is a really good tool, the spots, to count objects and to some extent for intensity analysis. But if you want to capture everything, you will have to go to surfaces, and that's what I'm going to do next. So I'm a surface guy. I like surfaces much more than spots. But the surfaces, they build up on the spots. So I do want to create a little story here. I start with the spots, you go to surfaces. But I would have just gone to surfaces right away, to be honest, unless I only wanted to count and get a quick count, nothing else. I don't care about anything else, then yes, just do the spots. But other than that, I would just go to surface, because even with the surfaces, you can also count. You're welcome. Any other questions? Okay, just quickly, one last thing before I go to the surface. So we have, this is the classification. So we told that, hey, you're green because you have this intensity, you're blue because you're dim, you're purple because you're medium. We force it into ranges. We also have the option to use a gradual heat map. We can go here into the color tab. And if I go to statistics coded, I can say I want an intensity mean of channel 2 heat map. So it takes the highest value and the lowest value, and it puts, a more gradual heat map rather than, hey, from here to here you're green, from here to here you're blue, and so on. So you can see like the dimmer guys are like purple, blue here, and the brighter guys are in the orange-red area. So it's more of a gradual distribution. Yeah, so I use this quite a lot as well. So I just want to show this as well. Okay, so let's go to surfaces. Okay, so I'm gonna now go into surfaces, and first this person wants to know the distances of each of those microglia to the vasculature. So we're going to do a surface of the vasculature, and then I'm going to also do a surface of the microglia to get more accurate statistics out. Okay, so first surface here, I call it vasculature, like this. And this time I'm not going to classify, because hopefully it's just one connected object. It won't be, because I have, stuff that is disconnected, but I don't need to do any classification in this case. Object to object statistics, yes, I would like, because I do want to know the distance of those microglia towards the vasculature. So going to the next step. So this here, it's going to be a little bit different. It looks similar here. We choose the channel. Again, I'm choosing this time red channel. Okay. And then we have different objects that we can, or different segmentation tools that we have available now. So first we have a smoothing, a surface detail that's different than we had before. And then we have thresholding techniques. We have 4 different thresholding techniques here, okay? So if you've used any version before 9.9, you would have seen 2 segmentation thresholds. Then the LabKit pixel classification was introduced in 9.9. And then the regular pixel classification, this is a new feature that is coming out in version 10.1 next month in November. Okay, so it's a little sneak peek here. more machine learning on the way. So basically, yes. Okay. I don't know. We don't know. It's part of the brain, but I don't know too much more of this data set in terms of like it's a co-culture and whatnot. But it's definitely it's a deep stack here. So we have deep, like it's I think I mean I have to look it up. But I think it's like really imaging deep into the tissue, and we get a few microglia here. But it's a demo image, yes. So you also have access to it in the demo IMARIS version.
You probably have to be kind of de-identified, or you don't have all the information that you use.
Exactly, yeah, exactly. And I do know who measured it, and I could ask this person, but I mean, this is a demo image, so I don't know all all the history behind it. Okay, so just a rule of thumb here in terms of the thresholding techniques. So the first two are the non-machine learning ones. So we use absolute intensity rule of thumb when the intensity of the objects of interest that we want to extract is homogeneous. So all of them are like bright, or all of them are like dim, or in the same range. Okay. Most cases, you know, you've seen your own data, like we will have dimmer objects and brighter objects, and they're all of interest. So there is a gradient in the intensity. So this is when absolute intensity will not work. And then we can use background subtraction, local contrast. This is if we have dimmer and brighter structures in an image that we're interested. Then the pixel classification, this is machine learning segmentation. This is if the data set is hard to segment, if there is, you know, the intensities are so similar between objects that you want and that you don't want, or things are so close to each other. It's just hard to segment. You can teach and train the software with machine learning techniques. So based what you guys already have available here is a lab kit for pixel classification. So lab kit is external software. We have the bridge integrated. So it's a seamless experience. We're not duplicating any data, but we're using external pixel classification and then bringing it back into IMARIS. So this is what you, if you've used IMARIS 9.9 or 10, you will have seen this. Now, in 10.1, we'll have our own pixel classification, which is also much, much faster than the LabKit pixel classification. And it's seamlessly integrated into IMARIS rather than going outside and going back inside. So this will come in, it's slated for November. I mean, things can change. So, but for the next release, we'll have this pixel. So those two are machine learning techniques, okay? One is the outside source one, and one is like the native IMARIS pixel classification. And the surface detail, this by default, this is 2 pixels because the software assumes, hey, everything that's 2 pixels or less, you're not interested in. Okay, it's not always the case, but this is like the default. Okay, so I can show you what it means. So I'm not going to change anything. Let's just go for it and see what happens. Okay, so in the 3D view, it looks quite good, I would say, the segmentation. Maybe you can see a little bit is under-segmented, but that's why we have the 2D view. And I can already see here, you see this guy here? This is a little bit too smooth for my liking, okay? So we're not actually nicely here. So this is where I go back and I change the smoothing. So I'm going from 2 pixels to 1 pixel now, which is 1. And if I go to the next step here, you can already see it's much, much better here, okay? We did lose intensity here, so it's not good anymore here. So we can drag the slider here and adjust it like this, okay? So I can do those things. If you can, I mean, avoid dragging this as much, because this is where you include human bias, and you may be doing it a little bit differently than you, those kind of things. So I rather click A again to go to automatic, and I go back and try to change things, okay? I mean, it's not always possible. Sometimes you have to work with the histogram, with the threshold. But if you can avoid it, especially now with machine learning tools, I mean, you can have more confidence in the result. So for example, I can try to do background subtraction. I need to include the smallest feature of interest here into my size parameter. So I can again go here and just find like a thin area like this, like 4. And then I can input something like 4 in here and try to see if this gives us a better result. than the before. But here it looks like we've even captured less. So again, I can adjust it, but then I would have to include again bias. So I go back and just sneak peek. I mean, LabKit pixel classification you have available. If we can discuss this and talk about it as well in when we do the one-on-one sessions. And then this is a very tiny sneak peek here. We can use the pixel classification native in IMARIS. It's still a beta, so things may be a little bit not perfect yet. So basically what we do is we say what is foreground, what is background. So we zoom in here, you know. For pixel classification, a good thing to do is to look at your actual pixels and not interpolate it. Remember, when we can go to preferences and check off interpolation, it's integrated right here in the wizard, so you can uncheck the interpolation data here to look at your individual pixels, because you want to paint on individual pixels and tell the software, hey, this is foreground, you know, and those pixel values are background, those kind of things, you know. You go in here and say, yeah, this is background, those pixel values are background, those are foreground, and then you click train and predict, and it does the pixel classification here for you, all of it, you see. This. And again, it's not perfect the first time because here it didn't capture some, so you just retrain it, okay? So it's like, for those of you who have kids, you know, you tell your kid, don't do this, and they will probably do it again. So you have to teach them and tell them repeatedly, maybe after 10 times, if you're lucky, they don't do it again. So it's the same thing here. You say, bad software, this should have been foreground, okay? And, you know, and this is background, something like this, and then you say train predict, and it's trying to do a better job here each time, okay? So you tweak it to go and do a better job. Typically 5 to 8 iterations will give you a great result, okay? But I'm going to go back, and in this case, just to make it as seamless as possible, I'm just going to use the automatic, and I just increase this a little bit like this. Okay, so here we have the option that was asked earlier to split touching objects. The things are overlapping, and I'm not doing it for the vasculature, but I'm quickly doing it for the microglia afterwards. So I'm going to go to the next step here. And here you can see, you know, I have my result, but I do have a little bit of quotation mark backgrounds, like those round guys, those guys, I don't want them. So I'm going to filter on volume, and I'm going to filter the small guys away. Just keeping those guys. I mean, here in this case, do you want to keep this? Do you want to remove this? So you can decide, you know, to keep those, remove those. Let's say this looks like a good compromise, and I'm going to finish it. And then now I have my vasculature and my microglia here. If I now go into my microglia statistics, here, I go to statistics and recalculate. Now I have a new statistic that I didn't have before that's called the shortest distance to surfaces equals vasculature. So I get the distance from each of those spots to the vasculature. So I can scroll from high and sort from highest to lowest. So I can say 72. I can zoom in. Which one is it? This guy. This is the furthest away microglia from all of them to any vasculature. And then I can scroll all the way down. There's negative values, minus 3.8. Why is it negative? Because it is overlapping with the vasculature. It is in here. So we can also quickly make the vasculature transparent here. Just see this guy is in here. Okay. But again, those guys are not like the microglia are not spheres. Okay, so we do want to have the actual surface, because here the distance is based from the center of the spot to the outside of the surface. If I have two surfaces, then it's from the outside of one surface to the outside of the other surface. So let's go back and do green, but this time as a surface. I'm just quickly going through it. I'm adding a new surface. I'm labeling this microglia. This. And green, and no classification, yes, object to object statistics, green channel. So this guy here is, for example, a good microglia. to show, okay, so if I don't change anything again, and I do next step, so this is the result. It's basically, it's a blob. It's not a microglia. You see that? So in this case, the surface detail, the smoothing was way too high, because if I go into the slice view and I get an approximation of my processes, they're like 2 microns, the processes. So having a smooth of 2 microns will just smooth all the processes away. Okay. So in this case, it's a case where I would turn off the smooth completely. And then I go to the next step, and you can already see how much better it's not a blob anymore. It's like this. Okay. Again, it's not perfect. You see, why is it not perfect? It's because the cell body is much brighter than the processes, much dimmer here in this case. So as I mentioned earlier, the absolute intensity is if your intensity is homogeneous all the way through, but in this case it is not. So if I use background subtraction instead, and I have to use the smallest feature of interest, which in this case are my processes, the 2 microns, then you can see the result is much, much better now. So now we have a very accurate result without even having to use machine learning techniques in this particular example. If I look at it in 3D, you see a lot is going on. It's going to pick up a lot of background as well. And there's a high chance that some of them may be touching or overlapping. So in this case, we have split touching objects. And what we essentially are going to use is we're going to use the spots that we created earlier now in the wizard. So we didn't need to create them earlier, but I'm using them in this wizard to split the microglia based on the spots based on them. Okay. And this basically creates an AND function. So only if we had something segmented and a spot is in there, it will stay as a resulting surface. So it also works as a filter. So our result looks like this now. Okay. And this is much more accurate than what we had before, where we had a sphere, because now we have the processes. Now, if we look at the intensity of each of those, it will take everything into consideration, all the processes. So I click this guy, you see this, and I go to its statistics. And if I go into the intensity now, intensity mean of channel 2, then this intensity basically is everything that you see here.
That's some average.
We also have sum, yeah. Oops, I did actually max, but the mean is just the sum divided by the number of the pixels, and the sum, we have the sum with the mean standard deviation. We have a bunch of statistics for intensity. You can also go to average values, and you can see here intensity, mean, and in terms of mean, you have the minimum, the max, the mean, standard deviation, median, the 25th percentile, 75th percentile, the sum, count, all those statistics you can also extract here from the image. But then we also have 2 new statistics that we didn't have before, where we had spots and surfaces. Now with surfaces and surfaces, we have the shortest distance. Again, we had this before, but now we also have the overlap volume ratio and the overlap volume. The overlap volume ratio gives us a percentage of how much of each microglia is overlapping with the vasculature, meaning how much of the green is overlapping with the red. So this is already the object-based co-localization. And the overlap volume will give us a number, how many cubic microns is the overlap between each microglia and the vasculature. You can also get a global count, how much of all green is overlapping with how much of all red, or how much of each microglia is overlapping with the vasculature. So all of this you can extract out of the statistics. Any questions so far? Yes.
So, is it possible to measure the cell body versus the branches? To separate them, not just the whole mean intensity.
So the question from the audience was if we can get the intensity not only of the cell body, but separate, like cell body versus, like, for example, here, this guy, cell body versus like the processes. With the surface, it is not possible, but with the filament object, yes, because in the filament object, you will create skeletonization, and then you can look at each of those processes individually. You get the intensity of each of those processes individually, and the cell body is treated as a surface, and you can get the intensity of just the cell body also individually. Yes. And I'm going to show the, as the end of the presentation portion, I will show a filament example, filament object example. Okay, so, oops. So I do quickly want to show the Vantage tool here. Okay, Vantage is the Vantage is where we do our graphs or plots in IMARIS. So, for example, we have 1D plots and 2D plots. I mean, this is nothing. Let's see, area notice, for example, and mean diameter, maybe mean intensity. Sorry. That's the mean channel 2. Just like a distribution here of one statistic, the intensity, or 2D view, we plot one statistic on x-axis versus one on y-axis, like a standard fare of making plots. But we do also have the spatial view here. This is where we can show a histogram count plot. So for example, we have the shortest distance to the surface and the number of spots on the y-axis, meaning, so 0 is the surface. So everything to the right are all of those microglia that are away from our surface. Everything to the left here are the microglia that are overlapping or touching the surface here. And we can see the number of spots, how many are there. And we can show this as a probability density plot to see in which region do we find most of our microglia. Like in this case, for example, from 0 to like 21 or so in this range, of distance, we'll find most of our microglia. And you can compare this probability density plot with a complete spatial random generated data. So the software calculates this random distribution for you, and you can compare the random distribution, which is the Gaussian curve, versus what you extracted from your real data. And you can already see that the real data is closer towards the surface than this randomly generated data, meaning there's some form of attraction. Something is attracting those microglia towards the vasculature, okay? And you can get this attraction distance here. This is where the curves overlap. And this is that you can use as an indication. Up to this point, you know, the microglia are closer and are attracted towards the surface, okay? You can extract this with the snapshot function, which is what you see is what you get. So you can extract the graphs as it is, like this. Or you can use the export SVG to export this graph as a vector graphic, and then you can manipulate and adjust things in a vector graphics program like Inkscape or so, for example. Then one of my favorite thing to do in the Vantage is in the objects view here. And I go to surfaces, so it's like a mini wizard. You see step 1 out of 2 down here. And you may have seen this in populations, but we have the gallery view here. The gallery view, oops. Vasculature off. You have to go back and turn the vasculature off. I just want the microglia. And I can use gallery view here. So we can plot all of the microglia on a grid, each individual object here. And then we have the option to sort them and color code them as well to our liking. So you may have seen this in some publications. So we can say we sort by volume. Then we have like the smaller guys here and the larger guys, you know, down here. And then we can also say, hey, we want to color code them based on, I go to the color tab here, and we can color code them based on position X. Okay, but we can choose any of the statistics to color code it and any of the statistics to sort it. Okay, and we can adjust the columns and rows and those kind of things. So this is one of my favorite things. And then the other is the scatter plot. So we can use a scatter plot. And for example, you know, with 2 dimensions, I would say maybe it's more like a gimmick, but you can say I want to plot the volume versus, you know, the shortest distance. And then you can see we have the shortest distance versus the, as I said, the volume. Instead of having just an X or a cube or something, you have the actual surface as a point, reference point. But I use this more to create more dimensions, like for example here. In this case, we have 4 dimensions. So you can create a quick plot where you have an X. The X is the volume, the Y is the shortest distance. On C, we have position X, but we could say maybe on Z we have sphericity. And then the color is based on position Y. So we have 4 dimensions in a quick glimpse. You can see this in one graph here. And then also you can export the statistics right from here. But it's just a way of visually showing 4 or 5 different statistics in one 3D plot. Any questions about Vantage? Okay, so I'm going to go back here to those guys. Okay, so I have the microglia, and again, in the microglia itself, I can also go to color code and statistics code them based on any statistics, for example, sphericity, how round they are, possible with spots and also with the surfaces. Okay. Okay, so any question about spots and surfaces before I move on to filaments? From the audience here from the Zoom session. Okay, so this was also like a demo data set for the filaments. I'm going to use actual user data. Any one of you guys in the room has used filament tracer before? Okay, so let me go out here. So we use this data, neuron, and this guy here. Okay, so this is the data set here, and I'm interested in this case in the red channel. So this guy here, the neuron, and if I go in here, edit image properties. So this is 1.2 gigabyte data set. Okay, so it's not super big, but it's also not like one of the smaller ones that we have in the demo data set. So it's 1.2. So I'm on a laptop. It's a more powerful laptop, but it's still a laptop. Okay, so if you have a dedicated workstation, things will be much faster, much smoother. This may crash. I mean, it shouldn't, but I'm just warning you, it's still a laptop. So maybe if some of you guys have used Filament Tracer on the Zoom session, virtually remoting in, you can see the difference. I just wanted to show you 1.2 gigabyte data set. I have a laptop. It's 64 gigabyte of RAM and an NVIDIA GPU. So I want to show you just how much faster it is compared to what you guys are used to, and also the machine learning tools in filament tracing. Okay. So first of all, if you do any filament trace, I would always recommend doing a surface as well. Okay, it doesn't hurt. If you want a complete picture of your data, it doesn't hurt to have both surfaces and filaments. Okay, so like we did before with the microglia, we did the surface, and maybe now you want to extract the intensity of the processes versus the intensity of the cell body. You can then do your filament, but you also have as a backup the surface data where you can get the total volume and you get the total intensity as well as a additional data. So it doesn't hurt. So I did it before. I'm not going to do the surface again here. So I have the neuron surface here. One time without those fine dendrites here. You see the small ones. And then I did a second surface here where I've included those guys as well. So it's up, what you want to extract. But let's say in this particular case, I do only want those. I don't want the finely guys. I just want those guys here. Yeah, there may be a question. Can IMARIS be run? The question is, can IMARIS be run on Mac or only on Windows? So both Mac and Windows are fully supported for IMARIS. Okay, so the surface is here. So one thing that is not explicitly written down anywhere is that you can use the surface that you've created to help you in the creation of the filament. Okay, so to do this, you have to make sure that in the surface you have the object-to-object statistics turned on. So when you start a new surface in the first step, remember, you can check it on. If you forgot to do it, you can always, once the surface is finished, you can go back here to the Edit tab, and down at the bottom is the object-to-object statistics, and you can turn it off here. Because this means now when we create the filament, it will use the information from the surface, because we turned this on, to fill the skeletonization with volume to create, the diameters of your dendrites in this case. Okay, so it's just a help. You don't have to use it, but it's a helpful guide as well. Okay, so now I'm going to create a filament object here. You see, this is the add new filament, the leaf icon. Okay, so if I click this leaf here, I have the filament. It is down here. So you can already see it's 10 steps by default. So a little bit more than what we've seen with spots and surfaces. And since IMARIS 10, it had an overhaul, so it looks different than if you've used before 10 and after 10. So you have 6 different options to choose from. So the first 4 have always a soma, like this little blue guy is a soma or a starting point. And then the other 2 here, those are like for vasculature, for example, or sometimes, you have a high zoom in here and you want to do a spine analysis and you only look at like the branch and there's no soma, then you will also use this guy here. So we have with spines, without spines, interconnection, no connection, no loops, those kind of things. So I can do a spine analysis here. You can see this, it's possible. I would recommend to do a higher magnification for accurate spine analysis than this, but again, it is possible doing it here. But I will not focus on the spine analysis. I will want to show the machine learning inside the filament tool for the segmentation of the dendrites. And I will use the first guy here, the first step. So we have a cell body, a starting point, and we have dendrites, okay, or filaments, but no spines. Spines would be this guy, and then we have 13 steps. I'm going to use this guy here. So I have to make sure that object-to-object statistics is turned on if I want to use the surface that I created before to help me with. So I turn it on. Sounds good. Next step. I choose the channel by default. I mean, it's the first channel here is good. And I need to choose a starting point. Okay. So this guy here, if I go back to 3D mode, this is my starting point, my cell body. So I can do it again as I showed you. I go to slice mode and go in here. And I can say click, something like this. And I have the 14.3 or something, Or again, I do it here with this cube. And it even tells you here with a pro tip. So starting with IMARIS 10 this year and all the future years, we want to include more pro tips here. So you don't have to, you're not lost or you have to read up on the help guide. So here it says when in pointer mode, use control middle mouse wheel. You see the same thing. This number changes based on the cube. And then I just zoom in here. I just say approximate size of this guy like this is 15.6. Okay. It's an approximation. It is not a magic number. If you do 15 or if you do 18 or something, it's all good numbers. Okay. Just a starting range. So if we go to the next step here, you can see it placed me the starting point here, and there's only one. So it did a good job, one starting point. Then we can choose calculate SOMA model or not. Yes, I do want to calculate the SOMA model like this. And then I can choose basically if I wanted to use a SOMA model, which is the surface, or if I want to use a starting point, which is the spot. Okay. So the difference is the length of the dendrites of the filaments is, if I use the starting point, is from the center of the spot to the end of the dendrite. And if I use the SOMA model, it's from the outside of the surface to the end of the dendrite. Okay, so just keep in mind, that's the surface, that's the difference. So here, you see this is the surface that was created. If you're not happy with this surface, you don't have like the possibility to play with it and change, the threshold and those kind of things in the wizard. But there is this pro tip that tells you can create a surface outside as literally as a surface like this, and then import it into your filament object. So that's a way. If you're not happy with what you see here, you can do it outside. You can even use the machine learning tool to make it as precise as possible and then import it. Okay. In this case, let's say I'm happy. That's okay. So I go to the next step, which is step 5 out of 10. So this is one new feature that we have in filaments that we didn't have before, which is multi-scale points. So in the past, it was just one size fits all. Now we have scale, because we have thinner dendrites, we have thicker dendrites, especially in vasculature. Sometimes we have microvasculature with really thick vasculature going on further down. So now we have the possibility to give a range. So we have thinnest diameter, and we have the largest diameter. So you could choose this. Big pro tip here, make your life easy, meaning if you see like this, the minimum is maybe 0.9, the maximum is 1.1, then don't use the range, just use one diameter. Okay, it's really if there's like a big gap. But if it's very close, like you can see here, I mean, there's not too much variation, unless I maybe take those thin ones, but As I mentioned, I'm not going to take those right now, but there's not much variation. So in this case, just use one diameter, make your life easier, okay? But you have the possibility. I do want to show, after I'm done here, quickly a data set. It's a light sheet data of a vasculature where we have to use the multi-scale points. Otherwise, we won't be able to get the tracing of this vasculature. But in this case, I'm just going to use the 0.9 here. Again, you can adjust those here with the pro tips based on the size of the cube, both for the thinnest and the largest diameter. I'm going to keep it as is. I'm going to go to the next step. What the next step does is basically places seed points, which are little spots like this, around my dendrites that will be connected later on to create the skeletonization. Okay. Two things of note. First, it will only trace until the last spot. Like it will here in this case, will trace this guy until here. So will not trace until here, it will stop here. Okay. So you can create manually, like it says in the pro, shift and left click to manually add or delete spots, the seed points. Or what I like to do is I use the slider, drag it to the left, and you can see this guy popped up. And then this guy popped up. Now it's going all the way here. This is not manipulating the data or like doing a bias, because the next steps are the machine learning steps where we train it, what is real and what is not. But here we only include, more spots that will help us later on to get a correct tracing. Okay. Like for example, this guy here, it's hard to see, this very dim here, but I would say this should be the whole guy here. So I do need some here because right now we'll stop here. So we'll put in a little bit more like this. So it goes all the way here. Okay. What also happened is while dragging the slider, we created like many more spots here. And then maybe if I do too much, it will pick up this noise here. So let's just force it to pick up this noise just to show you. See like this. If I do too much, maybe it picks up this noise. Okay. But I do want to do it on purpose because I do want to show you how we can eliminate this in the machine learning steps. Okay. If we have picked up something in the back, that's okay. Second thing to note is that we have to be careful with the spines. Okay. Like here. Here it would trace and it would go up to the spine and go down here. So we don't want this trace. So ideally, I can do like shift, left click, and remove those spines. Okay? Ideally, I don't want those. Okay? But it's very tedious, and I can go on and on to delete those. But we will use machine learning techniques to get rid of it in the next step. Okay, so I don't have to worry. I wouldn't worry about it too much in this case. So let's go ahead. So now this is step 6 out of 10. The next steps 7 and 8 are both machine learning steps. So you can, if you don't want them, you can turn them off here. And this goes down to 7 steps. But I do want to show, obviously, both of those machine learning steps here. Okay, so let's go to the next one, the first machine learning step. And you'll notice it's not instantly here, it's still thinking and progressing. But if you've used the filament tracer before, it's much, much faster. Okay, so it's done. And then here, what we can see, this is the machine learning interface. There's a lot of pro tips down here and a lot to read because it can be overwhelming, and maybe you forget in a week or two when you're doing it yourself. Just, this is very helpful here. But essentially what you have to do is you have to assign which ones is keep, which ones is discard, and then train it from there. Okay, so what you do is you go in here, you click this guy, you say keep, you click this guy, which is a spine, discard, this guy, spine, discard, this guy, discard, this guy, keep. Okay, you can see I have 2 or 3. And you want to do it for, obviously as much as possible, but around like around 20, 25, maybe 30. And then the rest will be, we click train predict and it does the rest for you. Okay. So also already this is kind of tedious, click and then assign and discard. So we have a new feature also that we released in IMARIS 10, which is the swipe select. So we go into here and basically I'm going to adjust the size of my circle. I hold down shift and I do swipe. Oops. I hold control, swipe, 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 keep. And there's also shortcuts for keep and discard. There's A and D. So you can swipe A, swipe D, and then you can make this much faster and streamlined, like all those guys, you swipe select, discard, And then remember those guys I didn't want. So I'm going to swipe select a few here, discard, swipe select a few here, discard, and then also I want to swipe select those guys here, discard, and those guys keep. Okay. I'm doing,
Question.
yes.
When you're doing the keep and the discard, do you have any tips or suggestions, like should they be in all over the image or different parts of the cell or bright or, you know, like, do you want them to be kind of different?
Yeah, good question. So it's good to choose like representative areas and representative spots. So if you can go to different places, it's definitely helpful. It's not necessary because you have different steps to train, predict, and make it better. But if you use, different areas that are representative, for example, here, this is like a dimmer area. So I do want to do something here. This may be a super bright area. I want to do something here. This will reduce the number of steps you have to do to get to the result. Yes. I mean, I could technically do 5 and 5 of each and do train and predict, and so it will take me a little bit longer, but it's also possible. But the recommendation is around 20, 25. And the minimum is what I've seen or experienced to get a good result in 5 to 8 iterations. So keep like a little bit more like this, keep, and then maybe like this, keep. Yes.
So let's say you're doing this for one image, and you said if you're happy with how it's trained, you can apply the same settings on the other images.
Yes.
So you don't have to do this all over.
Correct. Yes. So the recommendation is to do it. This will take some time. Both of the machine learning steps will take some time in the beginning, but then you will be able to apply it to multiple data sets and save a lot of time in the long run. If you just have 1 or 2 images, then yeah, you don't need to focus on that too much, probably. But if you really want to apply it later on 100 images, I would suggest spending a fair amount of time on these 2 steps just to make it as robust as possible. Then test it on the second data set and then retweak, of some of those, test on a third, retweak. And if it works for a few, 5 or 10, then I would be confident and apply it to as many other
Can you
images.
train this on like 2 different images of the same type? Like with an image 1 of whatever experiment you want, and you have an image 2 for the experiment.
I mean, not at the same time. So you do it once, you finish it up, you save it as a batch process, you apply the same one at the second one, and then do retweaking, and then you save the new one, and then you can apply this new one that's a combination of the first 2 on a third one, and so on and so forth. Yes.
There's another question.
Yes.
That are the dots that you keep marked in any way so you know which ones you kept. I wondered that too. Will it let you know if you've accidentally tried to keep something twice?
Okay, so because you don't see it too much here,
Right,
you can change the color to green, for example, like this, and now you can see those are the ones that I've selected. And even if you take this guy again here and you keep it again, it stays at 24. It doesn't add it, so you do not risk doing anything, in quotation marks, wrong. But then you can see those 2 here. Those 2 I can keep. See, now I have 26. So this the color. What was it? Was it? I don't even remember. What color was it in the beginning? Green, right? Or why? I mean, it was not a good color, the default one. So you didn't actually notice. Here, I mean, red. Yes, it's clear. But yeah, you can always change the color to something that you can see better, for sure.
And then they'll stay that way.
And then they stay that way. Yes.
Okay.
So now I just did yellow, and then I take those guys here and keep them yellowish or something. Or if you don't like yellow, you can also use like purple or something like this. Yeah. And then I hit train and predict. And now all of them change color. So now they're either purple or red. So the software did the training for me for all of those. Okay. Maybe purple is not the best. Let's just do yellow. Like this. And then you can see, did it do a good job or not? Like here, all of those guys, like what? No, this all should have been keep. And so should those, yeah, keep. And then you do train and predict. And you go on and on. Now it took this one. It's like, no, this one should have been this guy because this is fine, train and predict. So you can go small increment by small increment or do more. I typically like to do small increments, but I mean, some other colleagues of mine, they do more each step. So that's the way to go about it. And then you can go in and just check how well it did. Like all those discard, yes. Then this guy here is super weird. Why? No, that's absolutely wrong. It should be this guy. Try and predict. And then you can verify and take a look, and eventually you will get to the result. Like here, see, it did take those, but all of those, I wanted to discard this, and then also the spines, you have to take some time, as I mentioned, so it does learn and do all of those spines. They're going like, for example, this spine is good job here. But then if I zoom in here, this spine is detected well, this one is detected as a spine, is very good. Not too bad for sure. But there may be one or two spines here and there that it did capture, But yeah, it will take some time. I'm not gonna, do a perfect job. It's just to demonstrate you the workflow, the path here. So this is the first machine learning step. So I'm done, I'm happy. And I'm going to the next step, which is the second machine learning step. So this is also the most time-consuming step here. You can see, you follow the progress bar down here. This used to take a long time, and now it is much, much faster. And again, if you have a dedicated workstation, it will be even faster than for me on my laptop. Okay, so it's already done. And now again, by default, it took this color on. You can change the color at any time. You can change also this color at any time. You can see now it did connect those, and we have segments. So before it was seed point classification for the spots. Now it's segment classification. So it's the same procedure. You just tell them which segments to keep and which segments to discard. Okay, so you can see here did connect some of those here, but that's what I call the guitar strings here. But it knows that it's not right. That's why it's red. Okay, it does it because of the technique of how the algorithm works. So if all those red does, it's annoying for you. For how you see it, you can also turn it off like this. So in this way, you only focus on what is keep. Okay, you turn off all the discard, and looks pretty good. Yeah. So maybe you can just see here where there's some weird things happening, like here. This, there should be connections. You see, so you can bring in your red, and you can just tell the software this one should have been a keep. This one should have been a keep. This one should have been a keep, and then you do train and predict and tries to do better. And again, all the same thing here. You try to look at where did a good job and where not, which one it did keep and it should have been discard, which one they did discard and it should have been keep. It will take a lot of time again the first time, but it will pay off if you want to batch processes multiple times. Okay. So I'm not going to do too many steps here again because it will take some time. So I'm going to go to the next step here, which is step 9 out of 10. So we can here it's going to finalize our tree, our standardization. Basically, we can define a maximum gap length. So we can say if there's a gap of 1 micron or less, connect those. If it's more than 1 micron, then leave the gap open. And you can define those the gap size. And then you can finish here. And the last step here is let's see. I mean, you don't have to use the gaps. I mean, you can also leave it blank. Just go about it. But the last step that you do here is you have a filter of small little stubs. Okay, so maybe there were some spines that we weren't able to remove. So if I turn off the red channel, you can see this easier. You see like this guy here. Or this guy, those are probably spines that we didn't classify very well because we didn't take too much time. And by default, it says everything that's like 3.12 microns or less, it's going to be removed. And typically spines are not longer than 3 microns. But you can define the length and all of those removed. You can make it much larger, you can make it smaller. But this is typically like a good value. And then you can say, okay, remove those because I know those are not real here. And then you finish. And again, it's not perfect. You see there's some holes and things like this. Because I did a fast way, but this is the essential way how to do it. I've already done one of those before here. This one with spines even. And then I have this guy I've done also with spines, and then I have one without spines, this one here, that I've done, which is a better job because I spent more time. And then visualizing, you can do the branch points. You can visualize all of the branch points. If I'm in the right one, here, this one, you can visualize them. And also the terminal points, you can visualize here in green. And then obviously you can get all of those statistics as well. So if I go to statistics, there's a ton of statistics that are available with filaments. So you can see if I scroll all the way, it's so many. So you can really get statistics on the soma, just as a surface. So the intensity, volume, those kind of things. You can get the length and the thickness of all of the dendrites. You can get intensity in each of those dendrites. You can get how many spines per dendrite. You can get what is the shoal analysis, those kind of things, branch level, branch depth, a lot of statistics. You can always here go to the question mark and look up the statistics of filament, and then you can read up, what each of those statistics mean, how they're calculated. There's a ton of statistics for filaments. You can also do the same for surface and spots, obviously. But in filaments, there's just so many that you may want to just read up on what each of those mean. Okay. Any questions about the filament? Yes.
Can we color code like primary dendrites?
Yes. So you can go in here. Let me know if this is statistics coded. Like here. And then you can choose the statistics type. So you can say branch level. We have 1, it's coming out directly 1. Then 2 is where it splits. Like when it splits 1, it goes. So branch level and branch depth are the two main ones where you can characterize, primary, secondary, and so on. You can see here it goes up to 9, and they're color coded based on this. But you can color code on any statistics, like for example, the intensity as well of each of those dendrites, the most spines and those kind of things.
Are there different statistics outputs if you go to the vasculature method rather than just the original filament?
Yes.
So I guess my question is, can you just give us like a quick little difference if you were doing vasculature?
Yes.
You don't have to go through it, but
Yep.
that question in the chat further.
Oh, okay. Yes. Perfect. So yeah. So I did want to show you, if we go out, this light sheet data set here, which is a vasculature already done. Where is it? Oh no, it's here. This one light sheet. I mean, it's cropped as well, so it's not super large. It's just 13 gigabytes, but it's still, let's light sheet data set here. Okay, so we have, this is what it is, the volume. So here we have, thick vessels and micro vessels. You can see here. There's a lot going on here. So this you definitely could not have done before IMARIS 10, because one size would have a filament trace for a thick guy, so filament second object for the small. I mean, it would have been very tedious and would have taken forever because it's 13 gigabytes. So I have this guy here where I did one filament trace. Okay, so you can see, you can go all the way down to the microvessels and get individual segments. Okay, the segment is every time there is a branching point, this is considered one segment, and then you can get the statistics. Same, very similar to the neurons or like any tree-like branch structures. You can get how many branch points in entire. So it's called dendrite here because it's our nomenclature. So each of the dendrite is a segment. So this would be one dendrite. And then you can get the diameter of this, you can get the length of this. There's, option for spine and stuff in case it was a dendrite with spines. But in this case, I mean, if you do dendrite number of spines, you would get zeros everywhere for all of those in this particular case. But you have all those statistics available. Okay? The same set of statistics essentially. But as I mentioned, if you wanted to, you can go down here the bottom left where it says configure, does it say configure list of visible statistics, and you can turn off some of those. You know, if you're only interested in 8, turn off everything else and then use those 8. Depending on how you use, if you log in with your own password, then it will stay. But if you share a workstation without login, then just make sure you revert to default statistics at the end, so somebody's not like, hey, where's my volume? It's gone. But yeah, so you have the option to turn on and off different statistics as well. But there is a ton. But in summary, it's essentially the same batch of statistics that you have available for both vasculature or like any neurons or microglia. And in the past, those were treated very differently, and there was definitely more love for microglia and neurons. Now with IMARIS 10, they're all treated equally. They're all the same. They're all, yeah, basically the same in terms of being fast and treated equally. Yeah. So another question.
That was me.
Oh, okay. Awesome. Thank you, Jenny. Okay. I mean, any other question about filament tracing, filament? Good. So this is actually the main portion that I wanted to talk about in the morning. I mean, we still have 10 minutes if you guys want to see one little more example, or you have heard enough. I don't know what's your
Question on colocalization.
Mm-hmm.
So with localization, can we get results for localization of the soma versus dendrite, another protein? So basically having two different, that'll be for like an IHC with two different, there'll be two channels and then, yeah.
Right. Yeah, very good question. I mean, I have here microglia data set I want to show microglia. So basically the question was if we can get like colocalization in the soma versus like the processes and those kind of things. So the answer is yes. There's also a variety of different options if you treat it like as a filament object. So here we have, let's see, what do we have? So this, let me turn off everything. This one. Okay, so this is the data set. We have microglia and some protein. Okay, and you know sometimes it's expressed in the processes, sometimes it's in the cell body, and there's a ton of them that are like in a different plane as well. So the first thing probably in the workflow you would like to segment only the guys that are fully there with cell body and everything. Like getting rid of this guy because it's not fully there. So we can do this. So this is a surface, and then we can also get the surface of the protein or spots of the protein. Okay, so the spots you would do if you wanted to have a count, of how many of those. I mean, maybe sometimes you have like little puncta and you want to see how many of those puncta are overlapping. You want a number. It's going to be difficult, like in this area where like there's an agglomeration, or here, but it's possible. So you can do this here, and you can see now this one is 1, this one is 1, this one 1. Or I mean, you can just go to the surfaces here, and then you get overlap volume percentage surface area rather than just number. Okay, so these are the two ways that you can do. So I have done this here. And then on top of this, I did the filament trace. So here in the filament trace, I have now the soma and the individual processes. So just with the surface, you can only get the overlap between the whole microglia and the protein. But if you use this guy, you can get overlap and colocalization based on the soma. It's different, okay? I mean, and then you can use other tricks as well. So you can use the surface of the soma that you have here and try to subtract it from the surface of the main microglia, this guy, and just have a surface of the process. Essentially just creating a surface of the processes. So this is like let's say the more straightforward way, but you can also get a surface of just the process versus a surface of just the microglia, and then you get another set of statistics that are on top of that you could do of just the full surface of the whole microglia with its processes. Yes. And then in microglia, we can also do convex hull. I don't know if you've heard of this guy. It's basically just creating the box based on all the endpoints, and then how much of volume does this microglia take up in our data set. So this is also some nice thing that I typically like to do. Okay, any other questions?
Very nice basic question. So when you open an image and you do all of this, save it or it gets saved by default? When you open it next time, will you know everything you ever did to this image?
You have to save it, and then we'll know everything you've ever did. Yes, so definitely never forget to save. I mean, so you can do save, so it will override, or you can do save as. I mean, I typically start with one thing when I convert it and leave the converted default, and then I do save as, and I do underscore analysis or something. And it saves everything, all of those steps and everything. You know, if I go into this guy, the CD86, to the magic wand, and then here I have all the creation parameters that I've done. You know, it shows this number for this. I chose machine learning. I didn't choose machine learning. All of those things. And if I don't do it and I forget and I click Arena, it's asking me, hey, do you want to save it? So it's still gonna ask you, and then you say, oh, I forgot to save, and then you save it. Yes, you can give it a different name or something, and then it will remember everything that you have done. Yes. Okay, is there another question in the chat here? Oh, yeah. Awesome, thank you. Yeah, no, I mean, I think, I mean, it's a good thing, you know, just play with it, practice with it, you know, you have dedicated personnel at the core that knows what to do out. If they reach the limit, there's always the IMARIS support, so you guys are not alone, okay? So this is basically like a kickoff introduction. There's a lot we covered today. I think I will call it a day for the presentation portion. You don't want to overload your brain with even more information, but we'll just be discussing with some of you guys in this afternoon session for sure, okay?